plus, K plus pumps. Just out of curiosity, anyway, I won't ask you guys that either. Maybe I won't ask you that. It'll be a secret. Nice. The book talks about differences between animal cells and plant cells. So this is a good opportunity to just look at some of the transporters that are available. So if you look on the left side here, we have our animal cells. We see the Na plus K plus pump. This requires energy. In other words, it's coupled to the hydrolysis of ATP to ADP. Another example here. So this is a symport. Who can give me a decent definition of a symport? As decent. It's 8. I won't ask you much. No? No one? Symport. So we talked about it before. In this case, we're going to be moving a solute, probably against its concentration gradient, using the energetically favorable movement of Na plus into the cell. So the movement of Na plus into the cell is favorable because the concentration of Na plus is always higher outside of the cell than inside of the cell. So when it moves in, we can use that energy to move another solute with it. Okay, lysosomes. Does anyone know what a lysosome is? Yeah? For those saying yes, anyone want to get your 15 seconds of fame now? Digest different. Yes?
Something that like keeps the other organelles that are defective. Or...
That's right. Yeah, it can break down other organelles, proteins, sugars. It's actually a very versatile organelle. They're very much like bacteria in that they pump hydrogen into the cell. So this requires ATP, it pumps hydrogen into the cell. So we've all heard of the endosymbiotic theory of evolution. Who's heard of that? Yeah, everyone's heard of that. Come on. Basically saying that a lot of our eukaryotic cells today, as they were evolving, they took up bacteria that were carrying out certain kinds of functions. And as we evolve, we still have these bacteria today. What's an example of this? Our favorite organelle is mitochondria, right? That was probably some kind of bacteria that we incorporated into our cells to get energy, right? So everyone's familiar with that. There's a point to me saying that. I can't remember. Mitochondria. Anyway, it's interesting. It's pretty beverage. There's a plant cell. A few differences between plant cells and bacteria cells is that instead of, when they're making a gradient of ions, they're going to pump hydrogen rather than things like sodium and K+. They're generally simpler. And again, we're using an ATP pump to pump hydrogen rather than sodium and potassium. Okay, and again, why are we doing this? Why are we pumping things out? Because usually if they slide back in, we can use that energy to carry out some particular function. So in this case, whereas in animal cells we were pumping out sodium to use that energy, in a plant cell you might pump out hydrogen and use that energy as it comes sliding back in. Here we have a vacuole. What does a vacuole do? Yeah, it can store particular molecules. What about pressure? So what we're going to go into later is osmotic pressure, right? So if the water, the liquid inside of the cell becomes too much, the pressure becomes great and the cell can burst, right? Like a balloon. So some bacterial cells, or some bacterial cells or plant cells will have a contractile vacuole, which is used to control the pressure. So when we actually pump these things, we can change the flow of water into or out of the vacuole to prevent the cell from exploding. Okay? It's pretty morbid, you guys did that pretty well. So, passive transport. What we're going to talk about in quite a bit of detail, I suppose, is the movement of water by osmosis. Okay? The movement of water, as we said, it does not necessarily happen spontaneously across the membrane. It requires certain channels. We're going to see a guy who discovered that. We'll talk about the movement of other molecules via channels and transporters. Who can tell me what osmosis is? Is anyone? Osmosis?
The movement of water across the several hundred
Yeah, pretty well, semi-permeable membrane. As we said, that usually requires channels. So, I sometimes get confused with osmosis, or I used to, before I was a doctor. I used to get confused. I'm not confused. I'll start. Water the nature of water is that it's going to move to dilute the more concentrated solution. Okay? So if we have water here, there's solution A, whatever the solute is, it's at a lower concentration than the solution B. Okay? So this is more concentrated than that. So if this membrane here separating the beaker is semi-permeable, as I said, if it's semi-permeable, the water is going to move to dilute this. Okay? So everything in nature, because of laws of thermodynamics, everything's trying to reach an equilibrium. Alright? So if something is able to move, it will move so that the concentration here is equal to the concentration there. Okay? So you can define the osmotic pressure as the pressure required, or the energy required, to stop the movement of water from here to there. Alright, so it's represented in your book by this kind of diagram. You have to apply a certain amount of pressure to prevent the movement of water. Okay, we could call this the osmotic pressure, the hydrostatic pressure. Okay, now what do you call a solution? If I were to take a cell, the concentration of whatever solute in the cell is higher than the concentration of whatever's outside of the cell. That solution is said to be hypertonic, right? Hypertonic, someone said it. Hypertonic means the concentration outside is greater than the concentration inside. Hypotonic means the opposite, right? The concentration outside is lower, and isotonic means it's the same. There's an equilibrium. So if we were to look at a particular example, let's take your red blood cells, right? Because otherwise, who cares about this? If it doesn't happen in our bodies, some people do. But anyway, so we take our red blood cells. Difference between red blood cells and other cells in our body, anyone smart? Yeah, they have no nucleus. We pop out the nucleus, but otherwise they're generally the same as other cells. They have transporters to transport molecules in and out of, this bag. They're still a bag. They're not just like frisbees. They're containing stuff. And yeah, they work by the same rules as any other biological membrane. So if we're to put that in a hypotonic solution, what's going to happen? Why? So we say that there's a net water gain of cells. Why does this happen?
More protein in the cell.
More solute, right? We don't necessarily know which one. So what's interesting about this? It could be a whole bunch of different solutes. We could be talking about proteins. Generally speaking, it's ions that we're looking at. So the disequilibrium between cells is usually caused by different ions. And as we said before, it's necessary for the cells to cause a disequilibrium. Otherwise, we wouldn't have energy to do a lot of the stuff that we do, right? So normally things try to reach an equilibrium. The body goes out of its way to create a disequilibrium so that the flow of ions can be used to power certain functions in the cell. So in this case, the water is going to flow in. It's going to cause this to swell in a weaker biological membrane. It might just cause it to burst entirely and we get cell lysis. Now what happens if we do the opposite? What happens if we put this in a hypertonic solution where the concentration outside is higher? The cell will shrink. Why? That's right. So again, so the way I like to describe it is the concentration is higher outside. The water wants to move to dilute what's outside. It wants to make it less concentrated. So it's going to move out of the cell and cause it to shrivel like this. Right? So for those of you who like salty foods like me, you can imagine this happening to your red blood cells. I don't think this happens to your red blood cells. That's like a really high concentration. Okay, we have other examples such as, well, this is isotonic. Okay, what does isotonic mean? Is the water not moving?
Moving with that same rate.
It's moving in at the same rate that it's moving out. So it's isotonic, we have an equilibrium there. Are you going to get an isotonic situation in your body? Not really. The pumps are going to make sure that everything is kind of equal, right? Because the concentration of solutes inside the red blood cell will probably not be the same as in your blood. Plasma, okay? So the reason why we're not exploding is more because of pumps and things like that that are functioning to keep a particular equilibrium, right? Here's another example. We're just using a plant cell. What's one of the differences between a plant cell? So there's maybe two things in a plant